So while we get set up here, Dr. Zikánová is the scientist I was mentioning, the Zikánová database. She's been studying ADSL and related purine disorders for many, many years in the Czech Republic, and we're very grateful for the work that she's done over all these years on ADSL.
Okay. Okay, good afternoon again. And today I would like to present a short case report describing our recent findings of a new unsense variant in the ADSL gene. This variant leads to a very severe prenatal form of ADSL deficiency, and I will explain what this means and why it is important. Usually, when we talk about genetic changes, we use terms like missense or nonsense, and also like synonymous. And synonymous change is supposed to be silent. It should not affect the protein, but sometimes a change looks silent and still it breaks the function. To describe this, we use the new word for this, unsense variant. The case was about a 34-year-old woman from Germany who was experiencing her first pregnancy. There was no family history of genetic problems, and the parents were not related. And early ultrasounds and non-invasive prenatal testing results were normal, and first problems were seen much later. In the 23rd week of the pregnancy, doctors found that the baby was smaller than expected for his age, and they found changes in his brain structure and also in positioning. Unfortunately, the pregnancy had to be terminated, and many serious abnormalities were seen. The legs had almost no movement and were fixed in the abnormal position. The eyes showed structural changes, and there was small stomach and also cardiomegaly. And the brain and spinal cord were severely underdeveloped. And genetics tests like karyotype and SNP array and also routine exome sequencing looked normal. So the exact cause was not yet known in this stage. Afterwards, the parents still wanted to have a child very much, and of course it was important for them to understand what had happened. And they requested a more in-depth genetic analysis than the standard tests. And the exome sequencing data were re-evaluated and at the research settings. And it was how the new variant of ADSL gene was discovered. Here you can see the genetic results from exome sequencing. And you can see that, maybe, I don't know. You can see the baby carried two variants in ADSL gene. And he inherited a known pathogenic variant from the father, and it's a common disease-causing change. It's more common variant, and also synonymous variant from his mother. And as I thought, synonymous variants are typically considered benign, non-disease-causing, particularly when located within an exome, as in this case. Both parents are healthy, and because each carries only one variant. However, when the baby inherited both, the ADSL gene function could be impaired if the maternal variant proved to be pathogenic as well. Therefore, additional functional studies were needed to confirm whether the maternal variant was truly pathogenic. The case was referred to our team in Prague, which has been studying ADSL deficiency and other disorders of purine metabolism since 1993. Here you can see what happens in ADSL deficiency. As you know, when the ADSL enzyme doesn't work, two compounds, succinyladenosine and succinylaminoimidazole carboxamide riboside, accumulated in body fluids such as urine, blood, and cerebrospinal fluid. And we thought about it that they are probably toxic, especially Zicar. This accumulation is what we measure in patients' urine and blood, and it is a biochemical hallmark of the disease. And we have different forms of ADSL deficiency. Neonatal form, the most severe after birth. Babies have reduced growth, underdeveloped lungs, and severe brain damage, and unfortunately usually die very early. Severe form or type 1 form, children have severe psychomotor delay, seizures, and growth problems. And mild form, type 2 form, children may have developmental delay, autism, or seizures, but symptoms are milder. And recently was described also very mild form with isolated psychomotor delay. But in our case, we observed an even earlier and more severe prenatal form of the disorder. And the extreme severity is likely not primarily, it's my hypothesis, not primarily due to the toxic effects of accumulated intermediates as seen in other types, but rather to a deficiency of purines, which are essential for fetal growth and development. It is also highly probable that such prenatal cases are underdiagnosed. In the past, many pregnancies were terminated following severe ultrasound findings without any exact molecular diagnosis. And until recently, exome sequencing was not available to detect such rare genetic disorders. And consequently, many of these early and severe cases likely remained unrecognized. To determine whether the variant from the mother was truly disease-causing, we performed functional studies. And it means that we did additional laboratory tests beyond routine diagnosis. Although standard biochemical tests such as determination of SADO and Zicar in urine and plasma could not be done on the fetus, so we were able to study parent RNA. The RNA is the message copied from the DNA. And we also were able to measure enzyme activity in the parent's blood cells. For the RNA study, we first used a standard method to analyzing variants affecting RNA. We isolated RNA and reverse transcribed it back to the DNA and amplified it by PCR method. It's common method for testing DNA and sequence it. In the mothers, ADSL coding DNA, we saw just a normal variant, we couldn't see the synonymous variant. It was missing. And this finding contrasted with the father's ADSL DNA, where the non-pathogenic variant was clearly visible and consistent. It was consistent with genomic DNA. These results suggested that the mother's seemingly synonymous variant disrupted normal RNA production, although the exact mechanism was still unknown. We also measured enzyme activity in the parents' blood cells, and interestingly, despite being healthy carriers, both parents showed significantly reduced ADSL activity. It was surprising for us because ADSL activity had never been measured in carriers in our lab, and we didn't expect so large activities. We then confirmed these findings in the other healthy parents of children with ADSL deficiency, and it appears to be a common characteristic of carriers, especially with pathogenic severe variants. Reanalysis of exome sequencing data didn't show nothing new, but sequencing of parental whole RNA, which is a new expertise in our lab, provided the key answer. It showed that the variant causes production of short, you can see here, short abnormal not work properly and probably is quickly destroyed by the cell through a process called nonsense-mediated decay. That is why the maternal ansense variant wasn't visible in coding DNA derived from RNA, because the RNA mostly not there. To summarize, both parents had reduced ADSL enzyme activity, supporting the idea that there are carriers of pathogenic variants in the ADSL gene, and the mother's seemingly synonymous variant at the DNA level didn't produce normal RNA, and RNA sequencing showed that a shorter non-functional variant was generated and then destroyed. This explains why the fetus completely lacked a functional ADSL enzyme. The mother's variant was first classified as uncertain significance, and we didn't know if it was harmful. But thanks to functional studies, we could reclassify it as pathogenic. This means that in the future, prenatal and preimplantation genetic diagnosis is possible for this family and also for other families with this variant, because we uploaded our findings to public variant database, ClinVar. And this is I think the first description of the most severe prenatal form of ADSL deficiency. And it demonstrates that synonymous variants according to the genetic code can be pathogenic, and functional studies are essential for variants of uncertain significance. Enzyme activity can already be reduced in parents of affected children, which is new for us. And with this knowledge, we can provide families with clear answers and reproductive options. Also, in conclusion, with the right studies, even hidden genetic changes can be uncovered, and it giving families both answers and hope for the future. I want to thank my colleagues, our collaborators, and especially the family for their trust and for allowing us to share their story. And if you need any functional studies related to variants in ADSL or other genes involved in purine metabolism, or also for metabolite analysis and enzyme activity analysis, please do not hesitate to contact me at this email, and we can probably help you. And so thank you for your attention.
Wow. Dr. Zikanova, one thing I love about you is she'll write me an email that says, I just have a little presentation, a very small thing, and then it's like bombshell, bombshell. So I love that about you. Um, I'm sorry, can you, so I have a question here. Wait, where it is it? Where is it? Oh, it's darn it. It says, has anyone done substrate testing on carriers?
Yeah.
Now you have. I feel this
Yeah,
is,
but not substrate testing, because we know that the parents don't accumulate
Mm,
this. Yeah, we try it. We try it. Not I don't one or two families, not more, but we try it, and they don't accumulate. But they have very low activities.
that's very,
It's very interesting, I think.
yes. Dr. Chen is asking, Marie, is it possible to measure enzyme activity in blood?
Not just in blood, but in the leukocytes, or it's not just leukocytes, monocytes, leukocytes, and this type of cells. Not red cells, but not erythrocytes, but
Wow. Okay. Um,
I ask a question?
yeah.
yeah.
Yeah. Hi, Marie. I got a little bit confused with some of the numbers there. So could you kind of explain to me? It looked like you said 10% of splicing events were aberrant, but 40% of you thought you were losing 40% of the mRNA to nonsense mediated decay. And I couldn't square those. So
Yes, because uh, we were able to catch some copies of RNA, but we were not able to catch it with cDNA because of the PCR. So I think that whole RNA is degraded in nonsense-mediated decay, but we were able to catch some rest of this RNA.
Do we have any other questions for Dr. Zikanova? I have one, but it's just something I became curious about. You've been studying ADSL for how long?
In our lab since 1993, but for me since 1997 in my studies.
Okay. Wow. So do you ever get bored?
No, because it's always something new, and not just in ADSL, but with purine metabolism, and I think that it's exciting.
Yes. Oh, good. I'm so good. I do too. I wonder because I imagine you're in a lab and you're analyzing, and I just wonder, I hope this is keeping her energized. Exactly.
Yeah,
I
I
was
think it's
also,
always something new.
yes, absolutely. I want to just say thank you so much to this family too. Uh, I can't tell you how many times we've learned huge things about ADSL by just one family really, really pushing for answers for themselves. And I know that it can be painful and hard for a family to do, but I just want to say thank you to that family for exploring this so that we are all learning so much.